Benefits of GLP-1 Drugs Beyond Obesity by Daniel J. Drucker. Glucagon-like peptide 1, or GLP-1, is secreted from gut endocrine cells in response to food ingestion. It acts as an incretin hormone to enhance insulin secretion when glucose is present. Pharmacological activation of the GLP-1 receptor reduces glucagon secretion, which raises blood glucose and slows gastric emptying. This led to the development of GLP-1 therapies for treating type 2 diabetes. The GLP-1 receptor is found on several pancreatic islet cell types and in multiple regions of the central nervous system. Studies have shown that administering GLP-1 externally inhibits food intake through brain GLP-1 receptor activation in animals and humans, leading to weight loss. Decades of using GLP-1 medicines, such as liraglutide and semaglutide, for treating obesity and type 2 diabetes, have revealed other benefits. These include reducing heart and kidney diseases. There are several potential mechanisms behind these benefits, such as reducing systemic inflammation, which have implications for future clinical applications and drug development. The first approved GLP-1 medicines, like exenatide and liraglutide, needed once or twice daily administration. They were followed by longer-acting versions such as dulaglutide, exenatide once weekly, semaglutide, and tirzepatide, a glucose-dependent insulinotropic polypeptide receptor and GLP-1 receptor coagonist, suitable for once-weekly administration. A major non-metabolic benefit of GLP-1 therapies became evident in the cardiovascular system. Preclinical studies demonstrated that GLP-1 receptor agonists protect ischemic myocardium and preserve cardiac function after ischemic cardiac injury. These actions are independent of glucose control or weight loss. GLP-1 medicines have been studied in 8 distinct cardiovascular outcome trials in people with type 2 diabetes and 1 trial in people with obesity. Long-acting GLP-1 medicines, continuously present in the circulation, reduced rates of non-fatal stroke, non-fatal myocardial infarction, and cardiovascular death in people with type 2 diabetes and obesity. Trials showed a benefit for semaglutide in people with heart failure with preserved ejection fraction, with or without type 2 diabetes. How might this happen? Indirect roles for the cardiovascular benefit of GLP-1 drugs include reducing blood pressure and lessening atherogenic lipoproteins secreted from the gut, better control of blood glucose, and weight loss. However, preclinical studies show that GLP-1 protects the ischemic heart in normotensive non-diabetic animals more than what is achieved with weight loss. Furthermore, a long-acting GLP-1 therapy, albiglutide, which was withdrawn from the market due to modest efficacy for reducing glucose and body weight in people with type 2 diabetes, reduced the rates of major adverse cardiovascular events by 22%. Mechanistically, the distribution of GLP-1 receptor expression differs in the mouse versus the human heart, challenging the utility of preclinical studies for inferring underlying mechanisms in humans. GLP-1 therapies also reduce the development of atherosclerosis in sensitized mouse models, and clinical trials are underway in people with peripheral artery disease. The mechanisms linking GLP-1 receptor activation to the reduction of atherosclerosis and improved blood flow are not well understood, but may be independent of weight loss and are instead associated with reduced inflammation. Interestingly, the cardioprotective effect of semaglutide observed in people with obesity developed within months of drug initiation, well before significant weight loss was achieved in most trial participants. Furthermore, in the SELECT cardiovascular outcome trial studying semaglutide in people with obesity, the extent of weight loss did not correlate with the effects of the drug in reducing heart attacks, stroke, and cardiovascular death. Whether GLP-1 medicines might be cardioprotective in people with type 1 diabetes or non-diabetic individuals at high risk for stroke or myocardial infarction has not been studied. The actions of GLP-1 medicines extend beyond reducing glucose and body weight. GLP-1 attenuates inflammation indirectly through weight loss and neuronal GLP-1 receptor activation. and directly through GLP-1 receptor activation on T cells. It reduces complications by targeting GLP-1 receptors in multiple organs, originally shown to reduce blood glucose and body weight. Subsequent trials demonstrated that GLP-1 medicines reduce the cardiorenal complications of metabolic disease. GLP-1 medicines are currently being explored in a wide range of neurological and psychiatric disorders. Clinical trials and animal studies support a role for GLP-1 medicines in treating people with metabolic liver disease, and a phase 3 trial is underway with semaglutide. The GLP-1 receptor is not expressed by hepatocytes, challenging the understanding of how GLP-1 improves liver health. Although weight loss is associated with reduced liver steatosis and inflammation, preclinical experiments suggest that rare populations of intrahepatic GLP-1 receptor-expressing cells, largely endothelial cells and T cells, contribute to the therapeutic benefits of GLP-1 in metabolic liver disease. Intriguingly, semaglutide reduces the rate of kidney disease and cardiovascular death by 24% in people with type 2 diabetes, and gain and loss of GLP-1 receptor signaling improves or deteriorates kidney function, respectively, in animal models of renal injury. The understanding of the renal mechanisms of GLP-1 action is incomplete. Although sustained GLP-1 receptor agonism reverses diabetes-induced dysregulation of gene expression in multiple renal cell types, GLP-1 receptor expression in the kidneys localized to a subset of vascular smooth muscle cells, but not glomerular epithelial or tubular cells in the rodent and human kidney. Hence, whether the effects of GLP-1 medicines on the kidney reflect direct renal effects or indirect contributions from extrarenal GLP-1 receptor-positive cell populations remains uncertain. The heart, blood vessels, liver, and kidney contain populations of GLP-1 receptor-positive cells that might contribute to organ protection. Another potentially unifying mechanism of action for GLP-1 receptor agonism is the reduction of inflammation. The enteroendocrine L cells within the mucosa of the small and large intestine function as pathogen sensors, secreting GLP-1 in response to infection or sterile tissue injury. GLP-1 in turn acutely reduces gut and systemic inflammation in animals and humans. Populations of intestinal intraepithelial lymphocytes and some exhausted T cells are the predominant cellular sites of GLP-1 receptor expression in the immune system. The intraepithelial lymphocyte GLP-1 receptor is required for transducing the local and systemic anti-inflammatory actions of GLP-1 when inflammation is induced through activation of T cells. For example, using antibodies targeting the T cell coreceptor CD3. Nevertheless, the intraepithelial lymphocyte GLP-1 receptor is not necessary for the anti-inflammatory actions of GLP-1 when inflammation is induced by multiple toll-like receptor ligands, exemplified by lipopolysaccharide. Unexpectedly, the systemic anti-inflammatory actions of GLP-1 that attenuate toll-like receptor-activated inflammation require GLP-1 receptor signaling in the brain within neurons. Moreover, alpha-1 adrenergic and delta and kappa opioid receptor signaling are critical for transduction of the systemic anti-inflammatory actions of GLP-1 medicines. Brain GLP-1 receptor is also required for the actions of GLP-1 medicines to reduce systemic inflammation in the lungs and myeloid cells of mice with polymicrobial sepsis. Gain and loss of GLP-1 receptor signaling reduces or exacerbates, respectively, the extent of neurodegeneration and neuroinflammation in experimental mouse models of brain injury, stroke, or neurodegeneration. Several clinical trials have examined the therapeutic potential of exenatide in people with Parkinson's disease, with mixed results. A larger phase 3 trial is examining the potential efficacy of once-weekly exenatide in Parkinson's disease. Examination of real-world healthcare databases and clinical trial data link the use of GLP-1 medicines to reduced rates of cognitive dysfunction in people with type 2 diabetes. And two phase 3 trials, called EVOKE and EVOKE+, plus are assessing the impact of oral semaglutide in individuals at risk for progressive cognitive dysfunction. The expanding use of more potent GLP-1 medicines, such as semaglutide and tirzepatide, which produce greater reductions in blood glucose and more weight loss in people with type 2 diabetes or obesity, has led to scrutiny of whether these medicines might modify the outcomes of multiple central nervous system disorders. These include depression, compulsive behaviors, excessive use of alcohol or narcotics, and suicidal thoughts. Reports of psychiatric disorders in the SELECT cardiovascular outcome trial were not different over more than 3 years of exposure in people with overweight or obesity, and a history of cardiovascular disease randomized to semaglutide versus placebo. Analysis of rates of new or recurrent suicidal thoughts in 2 different real-world cohorts within large healthcare databases of people with overweight or obesity, with or without type 2 diabetes, revealed lower rates of suicidal thoughts in those receiving semaglutide compared to users of other glucose-lowering or weight loss agents. The relative chance of these events occurring was 0.27 and 0.44 for new or recurrent reports of suicidal thoughts, reflecting reduced rates in semaglutide-treated individuals. Moreover, analysis of electronic health records in the Trinex network revealed lower rates of new or recurrent cannabinoid use disorder in people with type 2 diabetes and/or overweight or obesity relative to use of non-GLP-1 glucose-lowering or weight-reducing medicines. Although anecdotal reports of reduced alcohol use with GLP-1 medicines are common, the results of randomized trials are inconclusive. Individuals treated with dulaglutide for 12 weeks with the primary goal to examine smoking cessation reported reduced alcohol consumption. However, people with alcohol use disorder randomized to exenatide once weekly for 26 weeks did not reduce the overall number of heavy drinking days, despite exhibiting less alcohol cue reactivity in the septal and ventral striatum regions of the brain, as determined by functional magnetic resonance imaging. Anecdotal reports of improvements in a wide range of dependence-related behaviors have prompted multiple randomized controlled trials to determine whether GLP-1 medicines might have therapeutic use in these disorders. The success of GLP-1 medicines for type 2 diabetes and obesity has fostered interest in developing next-generation therapies that are even more effective and produce greater weight loss than current GLP-1 receptor agonists. Tirzepatide simultaneously targets the glucose-dependent insulinotropic polypeptide receptor and the GLP-1 receptor, resulting in unprecedented glycemic control and weight loss. Like GLP-1, glucose-dependent insulinotropic polypeptide, or GIP, is also a gut peptide that is important for the physiological control of blood glucose. Pharmacological activation of the GIP receptor with a long-acting GIP receptor agonist also produces weight loss in humans, as GIP and GLP-1 exert complementary actions through distinct receptors. Simultaneous activation of both receptors provides an opportunity to maximize metabolic benefits beyond targeting only one receptor. Additional GLP-1 medicines under clinical development include those combined with GIP receptor antagonists, glucagon receptor or GLP-2 receptor agonists or amylin receptor agonists. The goal is to achieve greater weight loss while preserving or enhancing the cardiorenal and hepatic actions of current GLP-1 receptor agonists. These new medicines are often designed as peptides for injection and, in some cases, developed as small molecules or peptides formulated for oral administration. How might these emerging combinations improve outcomes in people with type 2 diabetes or obesity, at risk for developing cardiovascular, kidney, liver, or neurodegenerative disease? The receptors for glucagon, GIP, and amylin are all expressed within the central nervous system, but much less is known about their potential for neuroprotection relative to GLP-1 receptor agonism. These receptors are not highly expressed in the human heart, and their likelihood of modifying GLP-1-mediated cardioprotection has not been carefully scrutinized. Glucagon receptors are expressed in liver and kidney cells. Available data suggest that glucagon receptor activation may confer additional benefits, perhaps reducing rates of metabolic liver disease and diabetic kidney disease beyond what is possible with GLP-1 alone. although a short-acting amylin analog, called pramlintide, has been approved for the treatment of diabetes in the United States for 19 years. Amylin receptors are expressed mostly in the nervous system, and there are no definitive studies examining whether amylin receptor activation improves long-term health outcomes. A GLP-2 analog, called teduglutide, has been used for more than a decade to treat intestinal failure, and GLP-2 receptor activation may improve gut barrier function and reduce liver inflammation. However, clinical experience with GLP-2 receptor agonists in people with type 2 diabetes or obesity, alone or in combination with GLP-1 receptor agonists, is limited. The initial chapter of GLP-1 innovation focused on glucose control, and later, weight loss. Subsequent waves seem likely to improve health outcomes in people with a range of chronic disorders. Dozens of new molecules are being tested in the clinic, with some likely to target new mechanisms and achieve greater benefits in multiple disorders beyond more effective glucose control and weight loss. A wide range of clinical trials is underway, with results likely to support expanding the range of clinical indications benefiting from GLP-1 therapies. Hence, after almost 2 decades of the clinical use of GLP-1 for type 2 diabetes, and 10 years after the first GLP-1 medicine, liraglutide, was approved for weight loss in people with obesity, the next decade may bring even greater progress. This could introduce more powerful GLP-1 medicines while expanding the utility of GLP-1 therapeutics beyond currently established cardiometabolic disorders. Acknowledgements. DJD is supported by operating grants from the Canadian Institutes of Health Research, a Banting and Best Diabetes Center Chair in Incretin Biology, and a Sinai Health Novo Nordisk Foundation Fund in Regulatory Peptides. DJD has served as a consultant or speaker within the past 12 months to Altimmune, Amgen, AstraZeneca, Boehringer Ingelheim, Calliope, Merck Research Laboratories, Novo Nordisk Inc., and Pfizer Inc. Neither DJD nor his family members hold issued stock directly or indirectly in any of these companies. DJD holds non-exercised options in Calliope.